Dr. Dhruv, can you hear me?
Yes, I can.
Yeah, Dr. Archie is joining.
Yeah.
Everyone, the speaker is about to join and we'll just start after that. Just 1 or 2.
I think hello, can you guys hear me?
Yes, so we can
Yeah,
hear you.
so I think we'll wait for Dr. Chaitanya. I'm actually in my hostel room, so I cannot contact him directly. While we wait, can I have a +1 in the chat from the students, whether they know programming or not? So, like if they have previous experience with programming, can they just type +1 in the chat? So I just have an idea of how many students are familiar with Python or different programming languages. You can just type +1 if you know programming. This is related to the tutorial section later today. Okay, so I think we have only one person. Okay, so I think there are 2, 3 people who know programming. Okay, thank you.
So Dhruv, line is busy, is it?
Yeah, I tried calling too. It was busy. I'll call one of the lab members to check the
Yeah,
lab.
maybe in the main lab, if you
Yeah,
call, they can
yeah,
just walk around to his office and see what's happening.
yeah.
Meanwhile, I just want to talk to other and say how are you guys are doing, right? Are you guys following the program well? Do you have any suggestions, question at this point of time? If anybody wants to talk about their experience yesterday, I thought Sanjay gave a really good talk, and I think the student also was excellent in running the tutorials. In fact, I was feeling almost jealous that during my days we had to do it all by ourselves, trial and error, and things like that. There weren't any workshop like this, so I think it was quite useful. That's why we're recording it so that in the future, if people want to look at it, they can still do. Yeah, I understand it. I think all the participants are here. Yeah, yeah, Rachita, I'm happy you like that. The recording will help because I saw yesterday that some people, of course, when the tutorials go, people who are conducting tutorials will go at breakneck speed, and then others will be, can you go back two steps back and show to me again, kind of. So that's a bummer. And nevertheless, anyway, some of these techniques, I hope we may not be immediately using them, but it's good to know, and some of the keywords, keyboard shortcuts and things like that are very useful. One of the driving factors for conducting this workshop was that almost all biology now is becoming quantitative to an extent, especially microscopy, I guess. Nobody just looks at pretty pictures anymore. So that's one challenge. The other challenge is, I think towards the end you will see people do high content screens where we rake up a lot of images. It's almost impossible, therefore, then to go and look at those images and try and see what's happening. And so it's important that we are able to do uh, some kind of analysis without even looking at images. Maybe a day will come when you don't have any eyepiece. Microscope takes the picture and does some analysis and get to know. All right. Somebody says they can't hear me clearly. Is it same for everybody, or you can hear me?
We can hear you, sir.
Okay. All right. Let me try calling Chaitanya and see what's happening. Yeah, I'm unable to reach his cell phone. Not going through.
Yeah, he sent an email. He said that he's waiting to be let in. His name is not, doesn't have any permission issue or something.
So the the same link. I double check with him whether he's using the same link that was sent to you this morning.
Yeah,
If
I
he's
used the
using
same link.
something, yeah, you used. I know. I hope
Yeah.
he's also using it, because if he has used some previous link, it will not
Okay.
work. Yeah, he has to
Okay.
use the link that was sent today morning. Just put a note to him.
Yeah.
Dhruv, could you reshare your link to Dr. Chaitanya to the
Yes, I will
email
just
that he
reshare it
just
right now.
sent now? Yeah.
Yeah. Yeah.
Ravi, I would like to just announce to the students a small thing. So while Dhruv is trying to reach Dr. Chaitanya, participants, you guys can also, in case if you want to like to discuss and try to work for a few, half an hour or something, we are ready to create a breakout rooms. You can, if you want some additional discussion with the tutors, please let us know. Based on their availability, we can create breakout rooms accordingly. But definitely you guys don't give a long break. Try to go sit with the recording and try to work it in your computer. That will only help a lot.
Please join.
Hi, Chaitanya. Yeah, you have to unmute yourself. You're muted.
Thank you.
Hi,
Hi, how are you?
I'm fine. Good to have you on board.
Great to be here,
Nice.
finally.
Yeah, yeah. I don't know what was happening. I was trying to reach you on the phone, but I think the phone was also not going through for whatever reason. But we have been having this one or two small hiccups, with people are joining sort of and waiting for to be let in kind of a thing. But technically, the link should not have that issue.
Yeah, I don't know. Maybe I don't know. I don't use WebEx, but I know it's supposed to be very powerful. It's just,
Yeah, I mean, we are having so far so good. Let's see. I'll not. I would love to chat with you and catch up, but we'll hold up that later, and I'll get on with today's talk.
sure.
Is Ruchika going to introduce?
Yes, sir. I'll just start.
Go ahead.
So,
Ruchika.
good morning, everyone, and welcome back for the day 3. And today we have Dr. Chaitanya Atle from ICER Pune to give the first talk. Dr. Atle is currently an associate professor at ICER Pune. He did his master's in zoology from Pune University, then went on to do his doctoral work from University of Heidelberg, and did his postdoc from Heidelberg and MIT Harvard Medical School, Boston. His research interest lies in trying to understand the rules of self-organization in cellular patterns by looking at microtubule and motor interactions. And today he's going to speak to us about objective spatiotemporal dynamics of cells and organelles using computational imaging. So, sir, before we get started with the talk, there are certain instructions for the participants that will be given by Ritu, who, along with Tejal, will be coordinating the session with me. Over to you, Ritu.
So before proceeding, there are a few instructions I would request the participants to keep in mind. Please type your questions in the chat box. The questions will be taken up at the end of the talk. I would also request the participants to switch off their cameras and microphones at all times. Thank you. Over to you, Dr. Chaitanya. You may now start.
Great. Thanks. Am I audible?
Yeah, you are.
Thank you. So it was a pleasant surprise for me when Ravi wrote about this Images to Numbers workshop. And I'm going to talk about some work that hopefully gives you a general idea of particle tracking, which is, I think, the theme for today. And in the second part of the session, we're going to go over to something that is more hands-on in terms of how to actually do what we perhaps hope you will be able to achieve. Now, since there are a lot of you here, but I don't know exactly how many. I see. Okay. I'd like a very quick introduction from you with regard to your background in image processing. So from the participants, I'd like to know, have you used Fiji stroke ImageJ before? Okay, I've posted the question in the chat box. You're welcome to write into that, because as the instructions seemed to suggest, I think it might be a little simpler. Yeah. So all of you are familiar with Fiji. Yeah. Oh, wonderful. Oh, there's not a single no. Great. Have you used MATLAB before? Okay, so that appears to be majority says no, but I think there are a few yeses. What about Python programming? Okay, 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 okay. All right. Thank you for being so fast with your replies. So we'll try and take it slowly, but we're going to try and do something that, you know, like with swimming, they sometimes, once you have learned the basics, they throw you into the deep end. So we tried and throw you in the deep end, and we're there to help you, right? There are two of my students in the room. I think they will be guiding you through this. And as people who have recently learned to swim, I think they know what it feels like. So without further ado, let me just share my screen so I can start with the basic presentation I had in mind. And then we will switch to some hands-on sessions after a brief break. So just to remind ourselves, this is how I'm going to proceed. Just a second. Oh, having some computer issues. Give me a second, please. Okay, looks like it's back. And okay, right. Okay. Thank you. Probably. You probably now see my screen. So yeah, thank you. So for today, I'm going to broadly go about it in the following manner. We're going to cover some basics of tracking. Sorry, the pen doesn't keep up with my writing. Then we will go over an example of an algorithm or a workflow that we have developed as a case study and what we learned from it. And before I end this session for today, I'm going to give you a very brief, we can call it starter or a primer on Python. Okay, so that's the overall structure for today. I'm sorry for the handwriting. It's not as bad as it looks usually. All right, so you probably now also see my slideshow, I hope. Do you?
Yes, it's yeah.
Okay, great. So this is the topic for today. It is tracking cellular and subcellular mobility, and I am, as the organizer very kindly introduced, I'm Chaitanya Atle. I've been at IISER Pune since 2009, and just to tell you, because I think we are all in different places right now. I think some of you are in your home, some of you are at hostels probably, some of you are fortunate enough to be in your labs. We are located here in the southwestern corner, not very far from Bangalore, in IISER Pune. Oh, that arrow has moved a little bit. This is what our campus looks like, and it actually looks like that today also, because it's sort of rainy and pleasant. And my outline for today is really going to be broadly covering some basics of microscopy and sample preparation, digital imaging, computational image analysis, and then discuss some experiments and analyses we did with growth and mobility. And I'll briefly touch upon what chymography is, and then move on to single particle tracking and summarize. So the history of microscopy is quite an exciting one in my view. And perhaps if I asked you what the first microscope was, you should probably reply with one answer. It is a Dutch fellow called van Leeuwenhoek, right? Fine, just no need to do that. But as it turns out, in already 1595, there was a pair of brothers, Hans and Zacharias Jensen, in the Netherlands, who had built a two-lens microscope, essentially the first compound microscope. And they had presented it to a few people. It was presented to the French king. The French king was very fond of science and technology. This has been an important thing in patronage in most of the world with regard to encouraging science. And it worked, but not really well. And interestingly enough, the history of optics and understanding light and how we see things goes back to approximately 2,500 to 3,000 years, with the first known treatise, the first written material being the Optica of Euclid and Ptolemy's Optics. And in the area of what is today Iraq and Syria, Al-Haytham wrote a Kitab al-Manazir, which was an approach to refraction, reflection, and magnification. So theoretical aspects were there, but somehow building the devices took a lot longer. And then, of course, we come to this, which is a reconstruction of the original Leeuwenhoek microscope, which was around the 1600s. And again, some of you may have even seen this in museums, perhaps, if you've been fortunate, or even maybe in demonstrations. How many of you know what foldscope is? So you've heard of the foldscope being My question to you is, how many lenses does the foldscope have, if any of you have used it or heard about it? So one of you, Kiran N, answered 1. Khushbu says, I have seen people using it. No. I'm assuming many of you have though heard of it. It's quite popular. It's simple. It's supposed to be for a sort of low-cost microscope, which can allow you to do a lot of things. And the amazing part, the really amazing part about the foldscope is there is, in principle, in its concept, nothing different from a Leeuwenhoek microscope, 1600s. All right. So 421 years later, we are still using similar principles. I mean, I just want you to dwell on that a little bit as far as the hardware is concerned. Which also means in a way that we probably have moved much further from this and Robert Hooke's microscope. But something that becomes really important when we consider cells and imaging and tracking cells is, of course, whether you can even see what you're looking for, right? And so knowing what you know, that the Leeuwenhoek microscope had only one lens, whereas these two, both the Robert Hooke's microscope later and an earlier compound microscope, had two lenses, an objective and an eyepiece. Can you imagine which had the highest magnification? So there are three options. A is Leeuwenhoek, B is Hooke's microscope, and C is Jensen microscope, Jensen compound. I think the answer may have already come out because I have escaped my slideshow in order to be able to type in this environment. I don't know how to do both at the same time. I think maybe I can try this. This works. Great. So as it turns out, to probably, I hope, your surprise, certainly to my own, it was initially the compound microscope that had only a 3 to 10-fold magnification. Robert Hooke managed to achieve about 50, and Leeuwenhoek's microscope about 270 times. Let's just think about what that means, because if you recollect, something like an E. coli cell is a couple of microns, let's say 2 microns in size. So 270, if we are being generous, 270 can be rounded off to about 300. So 300 into 2 microns gives us 600 micrometers, right? 0.6 millimeters. Now, if you strain your eye and you look at your ruler, your meter scale, then you should be able to see 1 millimeter. So half a millimeter you can expect to see, which means that using the Leeuwenhoek's microscope, he was able to see bacteria. Yeah, I hope you recognize that this is 400 years ago we were able to see single bacterial cells. In fact, Leeuwenhoek was a textile merchant. He was trying to actually see the fibers of his textiles and realized that there's other stuff there. And this is one of those beautiful examples of trying to build something and finding something else out of it, right? So then there's a second aspect, which is the advances in sample preparation that further revolutionized it. So what you're looking at left is a page from the 1665 text of Robert Hooke, where he described the micrographs that he could observe and drawings that he could make based on those micrographs, meaning what he could see in his compound microscope. And then he also started using Leeuwenhoek's microscope because it was simply better. But it was not until the advent in the 1900s of a revolution in dye chemistry that you could start staining acidic and basic components of the cell and distinguish subcomponents and improve your contrast. Because remember, when you look at single cells, you somehow, and those of you who've tried doing this with just bright field, you know this, that getting contrast in a sample is not that trivial. The developments in optics since the 1900s have in fact allowed us to do a huge amount of both labeled and label-free microscopy. But labeling for a biologist, it's often one of the key things that you learn. You learn how to stain for nuclei, you learn how to stain for granules in the cytoplasm, and the red blood cell, or the so-called thin smear staining, is a classic experiment all of you, I suspect, have already done. And if you haven't, and you have access to a lab, I strongly recommend you at least try to find one of these samples, because they're beautiful to look at. Because they, in some senses, use the two core non-fluorescent dyes that we have been using for almost a century, which is methylene blue for DNA, RNA, and eosin Y for cytoplasm. And this is also commonly used in path labs, pathology laboratories. So when you go for your blood tests and so on and so forth. They these days, of course, use flow cytometers to measure any quantitative numbers, but for qualitative pictures, this is still a gold standard. So the processing of the sample, however, was through heat inactivation. And this heat inactivation, you can imagine that if you want to heat anything, yes, it will inactivate a cell, but it may also destroy its ultrastructure, which is what you're hoping to look for. So as far as light microscopy goes, for a very long time, optimization of fixation protocols was a very big thing. And methanol acetone or polyparaformaldehyde fixation has become the sort of standard for microscopy. And there are a few studies every once in a while which show that certain findings of shape, morphology, and so on cannot necessarily be made with the fixed sample. And then, of course, there is the last development that I want to talk about, which is molecular specificity. And the most obvious molecular specificity that we can get is with immunostaining. And antibodies have been developed now for long enough that you can label them in a multiplexed manner, meaning to say with labels that are non-overlapping, using the basic concept of fluorescence, and use this as a marker for the presence of DAPI. In the case of nucleic acid, it's DAPI, in fact, is the only one here which is an organic stain. All the others are antibodies against keratin, FOXP3, CD4, CD38, and CD45, and all these have become critical markers for diseases, and in particular in cancer detection. So the good part is that, of course, it gives you morphology as well as molecular information. The bad part is that it is based on fixation. And what you're looking at in the lowermost part is the fluorescence intensity, which is allowing you to detect the percentage of cells with a certain profile. And these are intensity histograms, which you may have already learned about. So what you are now going to see is a video that I think someone somewhere should have already shown you. But if they haven't, so those of you who've seen it, you can take a break for 2 minutes, for a minute perhaps. But for those of you who have not, maybe let me ask you, what are we looking at? So do you know what is the big bag-like cell? So Sajjan says macrophage. He's not very far. Anyone else? Macrophages. Okay. So since Sajjan replied with macrophage, can I ask you what is the difference between macrophage and neutrophil? Yes, phagocytic cell is a good general term, Sajjan. I agree. Yeah. So as it turns out, so I don't know, by the way, you may hear the videos. Do you hear the sound? Maybe best if I ask, do you not hear the sound?
So the sound of the video, right?
Yes, that's right. I'll play it and you just tell me if you don't hear it, all right? I'll play it, yeah.
Yeah, we can hear, but it's very low.
It's very faint. Yeah. So in that case, I'm just going to tell you what they're saying because otherwise it's a bit boring. So this is generally a neutrophil, and those of you who said phagocytic cells, you're not far. The little spots there, which were at the start of the video, here, these are Staph aureus cells. And the other balloon-like structures, as you might obviously guess, are red blood cells, right? And what happens during the movie is that the neutrophil is chasing the S aureus cells. Now, all of this is happening while the obstacles of the red blood cell occupy the space, right? And indeed, there you see sort of almost bleb-like motion. Yeah. And what you see at the end is, of course, they catch it. The neutrophil manages to catch the bacterium. And this is now sort of fed and ready, and now it's in replay mode. The idea is that these cells are working probably in the same fashion to track chemotax and move towards the targets, right? Now, this observational method, this idea that you can just directly observe something, record a video, and make sense of it, is very powerful, right? Because it sort of is this idea that if you see it, it exists, it's real. The problem with that, of course, is that it's just a description. So what do you do with just a description? You need to quantify it. And one of the tools that has combined the two things that I talked about at the beginning, molecular specificity and the mobility dynamics that you can see in a non-fixed sample, is the advent of the green fluorescent protein, right? And what you're looking at here is the classic beta barrel structure of the GFP protein with the chromophore embedded inside it and is highlighted in that right-hand side image, which allows at the wavelength of approximately 488 nanometers for the emission to be read off at about 510 nanometers, such that it will give you a specific signal. And because it's a protein, you can genetically encode it. And this development has become possible only in the last 20 years or so, ever since Osamu Shimomura was given a PhD project. I'm sorry, a master's project, I think. PhD, yeah. The project was the following. His boss gave him a sample and told him it glows, you find out why it glows. And And that was sort of as general and almost vague a statement as you can ever make. But that's probably what Nobel Prizes come out of. Very difficult problems and very clearly solved. What you're looking at on the left is the jellyfish itself, from which this GFP is extracted, and on the right are the excitation and emission spectra of the various proteins that are involved that have now been either found or engineered. And these three folks, Shimomura, Chalfie later on, and Rogesian, went on tweaking, improving, modifying these probes to actually allow them to become as powerful as they have become, right? So, of course, the limits are, there is no limit now to this kind of labeling, and so you can do combinatorial labels like this rainbow mouse with 160 colors with co-integration of tandem copies of transgenes that are tagged with these variations of fluorophores, fluorescent proteins, and imposing an RGB color scheme onto that gives you this beautiful spectrum almost, and allows you to then build a sort of brain connectome, and this is really exciting work because even now some of these techniques are being improved as we speak. Along with all that is the whole idea that you can also now combine two kinds of imaging, which allow you to both track cell morphology as well as molecular specificity. And what you're looking at here is the growth dynamics, of a bacterium, where the bacterial morphology was measured in DIC, and the DNA was labeled using a GFP tagged protein, which follows the dynamics of DNA replication through the cell. So in the smaller cells, you see little spots segregating, the red ones. In the large one, you occasionally see missegregation, and you can therefore maybe correlate both these. And the principle of differential interference contrast, when I said at the beginning that to improve contrast people develop dyes, was an alternative approach where you play with the optics of the light path using a polarizer and a prism before the light enters the specimen, and then after the object use a prism to further recombine the polarized and split light, which gives you the contrast at the eyepiece or camera, whichever you're using to view it. And I'm going to talk a little bit about it. That's why I needed to invoke this. And what is amazing is that with this kind of what appears to be relatively simple microscopy, even intracellular dynamics of single microtubule filaments have been reported all the way back in 1998. So microscopy has really made leaps and bounds of progress in terms of what you can do with it. And in some senses, it helps if we now take a step back and consider what we can do once we have acquired the digital image. Now, I trust that in the first few sections, I think this is day 3, so day 1 and 2, they've spent an enormous amount of time probably explaining to you what digital images are, what the acquisition process is, and what constitutes a digital image. So I think me talking about it is going to be redundant. If I'm mistaken in that, please do tell me. So, so maybe I will ask this first quiz question. What is a pixel? Okay, because that's the fundamental unit of a digital image. I would like you to type your answers in, please. What is a pixel? In other words, can you define a pixel or tell me the full form of the term, or is there anything that you can tell me about? What is a pixel? Picture element contains information in gray value, says Kiran. Good, yes. Super. Anyone wants to add to Kiran's answer? Photon for microscopy. Kajal, I think that is incorrect, right? Unit of a picture. Very good, Khushbu. Yes. Rachita says small area of illumination. No, incorrect. Okay. Go on. This is quite exciting for me. All right. Thankfully, there are no quizzes in this, but this is only for your learning. So my point of asking you this question was exactly to make sure that we are on the same page, that I hope you have reached a point that the next steps make sense. So a picture, as the first person, I think Kiran said, is a picture element. It's a short form, you may say. A pixel is a digital component, right? It's a digital element. Now, so my next question, yes, smallest unit of RGB value. Shubham, not really, yeah. It is, as Kiran said, it is a picture element. Kajal, it is not a photon, okay. We, of course, convert photons in order to get pixels. And so now maybe I do need to make a little detour over here and go back to my whiteboard. So my question to you was, what is a pixel, right? Okay. And maybe the corollary question, a continued question, is how do we get it? Because I think that is what some of your answers are sort of obtain it. Right? Now, a picture, a digital image, all of you know this, and it's best to demonstrate it rather than talk in theory, is made up of components. Now, what are those components? So someone said RGB and so on and so forth. So I'd like to point out that in its core, right, in its basic amount element, even if you take your beautiful cell phones and their cameras and the wonderful images that you get from them, they're using the same principle, right? You see that this image, I mean, let me pull up a nice picture I took recently. This image of buffaloes that you can see on the hillside. Now I'm going to do something that this doesn't allow it. Okay, so all right. Okay, since my demo images are not opening, I'll use something. Okay, great. Okay, so I think all of you see my screen. Yeah.
Yes.
Yes.
It's visible.
Okay. So what you're looking at is an image of taken in the lab by Shivani from my lab of a microtubule. This is not the best image that I could pull out. What you see when I keep zooming in, by the way, what am I doing? I'm zooming in. I'm using Fiji, which you said you're all familiar with, and I'm happy to hear that. What do you see now? You're looking at exactly individual pixels. Yes. That means each of these elements here, this guy, this guy, this guy, this girl, whatever you want to call it, they are all pixels. Now, my next question to you is, can you have something more resolved than a pixel? Can you have something more finely resolved than a pixel? The answer is yes. 1, yes. 2, no. 3, don't know. Okay. Those are the only 3 options I'm going to take. Can you resolve an image beyond? You see, I'm going plus plus plus. I don't know if you understand this, but I'm in Fiji and I'm clicking the magnification tool and I'm going plus and minus. So zooming in, zooming out. So my question is, can I zoom in further than a pixel? Khushbu says don't know. Okay. You know, you all have, I mean, I think most of you have some computer in front of you. Am I correct? So you can try it, because this is an experiment. It's, you know, computers are wonderful because you can try virtual experiment, and unless you format your hard drive, nothing can go wrong. So try it, please. Okay, it's important. Just try these experiments. Other than Khushbu, anyone wants to hazard a positive answer? Yes or no? हाँ या ना में उत्तर दें. She was brave enough to say, I don't know. I think it's a very good answer, by the way. So the bottom line answer, I'm still waiting. Shall I give you a minute? Okay, I'll give you a minute. I have received 21 messages, but I don't see them. Oh, I'm sorry. Okay, 2. My bad. Sorry. I'm so sorry. The scroll button wasn't visible to me. Okay. No. Wonderful. So all of you have answered 2 are correct, right? And that is exactly the point I was trying to make, which I hope reminds you that, as some of you very nicely said, a picture is the unit, the smallest unit of a digital image, a pixel. Yes. Which also means that now we can ask the next question: what does it consist of? And again, I think you have already covered this. So at the cost of being redundant, I'm going to draw a line across this, yes, and ask Fiji to plot me a line profile, yes. And what you see here, some reason the values show up as negative. I'm not sure why. Slightly weird. Okay, let's check. Oh, that's probably why. Okay. So
It could be because of 32-bit image, Chaitanya.
yes, thank you. So when I now convert this to a 15 and 8-bit image, I have now a range of values which are in a range of gray values that you are familiar with, I suspect, right? So this bit depth business, I think, must have also been talked to you about. I think that was Lakshmi who probably was adding. She probably has dealt with all this already. So great. So you are aware of all this, which also means that when you have pixels and you have gray values, your digital image is somehow taking the packets of light, yes, your photons that were impinging on your sample, yes. So you had a nice cancer cell here, so migratory fibroblast for all I know, and by whatever measure, whether it is through reflected mode or transmitted mode, you somehow pipe the light and put it onto your camera. So your camera, if it is a charged coupled device, CCD, right? Not Cafe Coffee Day, then it has indeed on it a grid, a lattice grid, which is going to capture the information in a manner that, depending on the intensity of the light, there will be more or less light falling on it, and therefore it will cause a more or less charge to be formed, which will in that case be turned into a value in your image sense, which finally gives you the gray value image. Okay, and that gray value image, depending on the bit depth that you have chosen, will have some intensities, and somewhere here it will be. So actually, I should probably correct this. If you had more light falling there, it will be brighter or lighter, you may say. Oops. So let's just rub this out. Forming, hopefully, what you will see as the outline of your cell. Yeah. Now, depending on the exact method of contrast, that will decide what is going to happen here. So let us just say for the moment, you had a cytoplasmic dye with a GFP in it, a cytoplasmic GFP. Then you will see bright signal here, and everywhere else, and you are familiar with this, you will see dark black or gray or some background, right? And this is something that is critical for all of what we talk about in the next section. All right. So this is basically your image rendering. Now, when some of you, I think one of you said that a pixel is an ROI. An ROI is nothing but a region of interest. In other words, if I now want to claim that I only want to focus on this segment of the image, this is an ROI, region of interest. This can be either done by zooming in on the CCD chip itself, so I only image this part, or by digitally, in other words, in the software, either acquisition software or in the image analysis software like Fiji, by providing the coordinates for the ROI. But pixel and ROI are not the same. Yes, that is what I wanted to really emphasize, because I know that not all of you think that, but it's important. Yeah. All right. Okay. But pixel is the unit, the smallest unit of your image. So, which also means that photon is not equal to pixel. Yes. All right. So I hope that is fairly clear. And let me go back to my slides and we'll continue where I left off. So now let us say you have acquired an image time series. Yes, let's say these are fluorescence time series. These were nuclear labels that I had acquired long ago as a PhD student using histone H2B markers with CFP, I think, cyan fluorescent protein. And when I did that, and these numbers are taken from more recent data that I have been taking, one single image, 512 by 512 pixel with 12-bit depth, gave me about 1.5 megabyte storage space required, right? Just 100 frames. Let's say I took them every minute, so 100 minutes, gave me 150 MB, obviously. And a Z stack of 20 steps in addition at every time point gave me just 3 GB, straight away 3 GB of data from just one single experiment. This is obvious. This is, I think, I'm sure Lakshmi and all the other people who've spoken before me have already mentioned this. This is the real crux of the motivation for computational image analysis. There is too much data, it's very large, and analyzing it manually is almost impossible at some point. You can do it for small data sets, and we've done it in the past, but you need to build something more than just that. So the pipeline that you need to follow for this, for particularly tracking that we're going to talk about, can, of course, first if it's 3D data set, then maybe you want to render it. You want to look at what it looks like, right? And this volume rendering, VOLREN as it's sometimes called, is just simply taking the intensity data and converting it into voxels. So like we had pixels earlier, we can also talk about voxels, which are nothing but, oops, sorry. The volumetric picture elements. So here we had pixels, yeah, and now we have voxels. Something that you are very familiar with is segmentation, but I guess I didn't want to go that far yet. When you have such a time series, there is a problem, and many of you who have done microscopy know this, which is that as you go through your time series, you will find that the sample may move, not the cell that is moving alone, but the whole sample, the whole cover slip or the frame of reference, let us say the medium, there is a flow. So how do you correct for that? Now, perhaps some of you have already done this, because I think I'm sure Lakshmi has been talking about a whole lot of methodologies, and I saw the program included some things. Does anyone have any idea what you should do? Let us say if you're using Fiji, how can you correct for what we call sample drift? Or how have you, any of you who have experienced this, corrected for it? Okay, Shalini says that this has been a big problem in the lab. Wonderful. Somen says Picasso software. I have not heard of Picasso software. I have heard of Picasso, but Picasso software. Okay, Shalini says instead of resolving it, we try to reduce drift. So I'm assuming you mean experimentally, right? You used to check and correct it at the time of acquisition, not used Fiji. Yes. Okay, great. What usually ends up happening is that you sit at the microscope and tune things, right? You move, you correct manually, and the brain is quite good at this, so maybe you can do it. The problem with that is there are many problems. Yes, stack reg, wonderful. Rita Bhas says stack reg. So exactly this is what I was referring to over here, which is called registration. Now, stack reg is an option in Fiji which registers a stack. What do you think? So let's say if you didn't know anything about stack reg, the most simple registration method is the fact that you assume that all your objects are what are called rigid objects, right? And any transformation is either a rotation, a translation, or maybe and that's it. Maybe in and out of focus is problematic because that is optical. So if you have rotation or translation, then by simply putting in place a comparison from frame to frame into the software, you can correct for what you may call spurious drift. Yeah. Now there is a slight problem here, which is that if you're trying to measure mobility and you perform stack reg or what is called then rigid registration, you may actually end up losing the information that you were trying to get out, right? So you understand that there is an issue. And there's a second problem, which is that, and that was what I was about to say earlier, that the modification in the object that you're following, let us say it's a cell, may not necessarily be just rotation or translation. It may also be deformation, right? And that makes registration quite complex. And there are many sophisticated techniques that have been developed, and StackReg includes some of these algorithms that use what are so-called landmarks. Now, once you say you have landmarks, it means that you have to sort of define those landmarks. So often, I think to answer Shalini's point, one simple way to solve this is to dope your sample in order to, at the time of acquisition, to reduce drift, is to put some fixed markers in your sample and then correct according to the movement of the markers, because those markers are passive. Either they are beads stuck on the cover slip, or they are objects in the gel that you're observing if it is a solid medium, or there is some way that you as a user know that some parts are not supposed to move, and that is what you will feed into the Fiji plugin like StackReg or something. Okay. Now, we said we are going to do talk about single particle tracking, but there is an alternative, and that is called flow estimation. And this flow estimation says that, look, I can't distinguish my cell from my background or objects from one another. I assume indeed that my motion is not subject to drift, meaning to say every movement is bona fide. Or if it is not bona fide, I can correct for it. At least I have a way of figuring out. And finally, I use this to take a point-wise matrix of all coordinates of intensity and look at intensity changes at that position. Now this comes back to what I was saying earlier, which is that what is a digital image, right? And in that context, so let me see, just add one more. Okay, there we go. Your image, oops, since we said is nothing but a matrix. So once more back to high school, right? And I think again, all the people, the speakers who have come before me have probably already spoken about it. So I like to call them J, I think most people conventionally call them, and rows, some index I. Then for every position I, J in time t, t1, t2, etc., if the intensity ij at t versus the compared to the intensity ij at t1, t2, t3, keeping the ij coordinates the same, show me any change, then I already have a modification, a change in time. Now, in addition to that, I can look at I plus 1, I minus 1, j plus 1, and minus 1, which means that I'm essentially asking the question, if this is my pixel of interest, yes, which has coordinates I, j, I plus 1 will just simply be this one, I plus 1, where j is constant, I minus 1 is this, j plus 1 is this, I'm sorry, j minus 1 is this. We go left to right in India, and in most parts, Arabia, you go right to left. Japan also, j plus 1. Okay, this is a convention we use, right? So, which means that knowing the, we can say, gradient of intensities in along rows and columns, we can tell whether the intensity has increased in this direction, or this direction, and by what amount. And using that, we can judge globally what is the flow. So this is what I meant by flow analysis being one possible way to measure mobility without doing anything further, right? And this is quite popular in engineering physics, engineering and biophysics, where people use something called particle image velocimetry, and I urge you to go back and look at some of the papers on that. I'm not going to deal with it in the interests of time. I guess I should probably at this point ask the organizers, I know we started about 10 to 15 minutes late. How are we doing with time? I don't know who is the host.
Yes.
Hi,
Hi.
I can't hear you. Oh, Lakshmi is the
Sir, you
co-host. Maybe
asked
she can tell me.
first.
Oh.
So we have 15 to 16 more minutes.
Okay, okay. I will speed up. Thank you very much. Yeah. I have been conducting this as more a discussion and an interactive class session rather than a standard seminar. So I would like to eat into my Q&A time. So I think it's okay. Yes. Does that include the Q&A time? The question session that I think was kept at the end.
No, sir. We have 10 more minutes for question answering session.
Okay, thank you, thank you, thank you very much. Yeah, great. So to continue, you can of course segment. What is segmentation? In the most trivial fashion, it is saying that if I have some pixel intensities, and you remember I drew a line profile, so let's bring up my Fiji image again. Right, so I think you see my intensity, my image. I again draw a line profile, this time across the whole image. I think I should probably convert it to something simple because 32-bit gray value. Okay. Okay. Now I see that, so maybe better still, why don't I do this? Right. So I have an image, and I know from the point that from how we acquired this image, that that the slightly brightish regions in this center are probably my filaments of microtubules. This is what I should see a line profile for here. And I see something, but it's not very clear. So one of the first things that I must do is to process it to subtract background. And obviously a 50 pixel rolling ball radius background subtraction was too dramatic. So this is not helping much. So perhaps I need to just simply do some smoothing of the image. And all of this, I think you have been again probably spoken to about this, is the process of pre-processing of images. Because without this pre-processing, very little from these kind of noisy images, very little sense can be made. You now see that things are a little bit less jagged from what they were earlier. And my approach now for segmentation, not necessarily in such ambiguous data sets, is that if I find that my filament intensity is highest here, and this is my position along X, so I drew a line profile, right? And my intensity is here, then by setting a cutoff, right? We can call it IC or threshold, which I think you're familiar with. Then I can basically decide that this is my object and this is my background. The resultant image will then simply be, if this is a 0 value, then something that is straight and a peak and something like this. So we'll call it 1. Which now means that in an image itself, a binarized image, which I will have got, will have a shape that very distinctly defines the object I was looking for. And that is what is the primary task that you try to achieve with segmentation. Now, after segmentation comes the next step, which is single particle tracking. Now, this particle tracking business assumes a few things. It assumes that from frame numbers as they change, yeah. So you remember I said T1, T2, T3, so I will use the same language, T1, T2, T3, T4. The object that I'm interested in, let us say it's a filament, is growing and shrinking in a way that I want to follow it. Yeah. So I need to, in order to know, to be able to do this, I need to attribute the filament to its cognate filament from frame to frame. And the most straightforward way to do that is to use the fact that if I now consider along some one plane of the image, one-dimensional section of the image, you may say, and I have multiple particles, yeah, and they're moving around. So they move like this. And I am interested in attributing each one correctly to their own cognate partners. By I can perhaps tell, but the computer doesn't know. Then I need some objective measure. And that objective measure is based on this basic idea that if I now draw a straight line for each position from the initial point, then I can use a cutoff radius or a search radius. Oh, sorry. Within which region I expect to find my object. Now, object finding is often reduced to finding the centroid of the object. So if the centroid is here, and if the range that I give it is sufficient, wide enough, meaning to say some distance r, then typically, we are talking about 2D images. So let us say I had a point here and it moved here from T1 to T2, then that search radius should encompass that same coordinate or the centroid in the second frame. If it does not, then that means I have not successfully joined them, which means that it comes down often to defining the so-called R value. Yes. So R we call search radius, and this essentially allows us to do what this graphic is trying to depict, which is that it follows multiple particles and connects them. Now there is a possibility, and this is an obvious possibility, which is that there could be more than one particle that may be within the search radius in the next time frame. So if you have no other information, how do you actually link, right? This is a tricky problem, and this has been solved by many ways. Most obvious is some kind of prediction, and Dhruv is going to discuss this in the hands-on session a little bit. But for now, I'm going to leave it to say that there are some things that we need to add to it if we want this single particle tracking to work. Of course, once you have this, you can also do other things like in a certain region that you have followed, measure intensity and concentration, which gives you the kind of trap and flip diffusive kinetics, or look at surface rendering, which is different from volume rendering because you're detecting a surface in 3D. And this allows you to quantify parameters like volume, stretching, velocity, diffusion constant, etc., right? So in the last few minutes available to me, I'm going to talk to you about a problem that we've solved in the last few years, where we were looking at the development of a C. elegans embryo. C. elegans N2 is the so-called lab wild type strain, and the fact that during the first embryonic division, which is asymmetric, there is a period of oscillatory movement, so back and forth movement, up and down. Now, one of the ways that we thought of trying to follow this is to follow the birefringent granules. Now, many of you know what differential interference contrast microscopy is, and I talked very briefly about it at the beginning. But in essence, it is label-free. I talked so much about fluorescence. This is not fluorescence, right? These lipid-based granules have a very high contrast in DIC microscopy, and they're very packed. So we decided to look at the image profiles of these, and based on the image profiles and a convolution with a filter, which is the part where we enhance the contrast of those structures, we could actually fairly reasonably go from an ROI, a region of interest, which I'm showing you as a demonstration, filtered image, which gave you such strong contrast that then we could very easily do black-white thresholding and get centroids, or the center of mass, as I told you earlier. And now, once you have all these beautiful center of masses or centroids noted, we can, of course, use the kind of distance metrics. But before we do that, we really wanted to know how good is our filtering approach with respect to some of the input parameters. So we use the filter size, K size, the variance, the sigma, and the strength of the filter, which is phi. And all of these seem to show that within a certain range, it's optimal. And we measured this in terms of sensitivity, precision, and F1 score, which are based on the comparison of the ground truth. Now, those of you who have considered image processing know this perhaps, that ground truth is something that we say that this is the actual position of the object, right? Because you're doing single particle tracking, remember. So after all, how do you know you're doing this right? And so the simplest way that we do this is by saying that, look, we can't analyze all these time series by hand, but at least for a few frames, we can analyze and manually detect. And now manually detecting means I find this is the center of the granule, the other person finds something else, and the third person finds something else. So we try to average across people. And this is our ground truth. Now we compare our algorithm to our ground truth, and we get false positives, true positives, and false negatives. This allows us to, of course, calculate metrics like sensitivity, precision, and F1 score. And this tells us that the different algorithms that we compared, there are some that outperform the others. And the positional detection error is something we want to minimize. Gaussian filtering was not that great for this data set, and you may find that in many data sets too. Either way, the outcome of that was this beautiful looking movie that Yash and Dhruv generated from the C. elegans data, where you can now see that while the mobility that I was mentioning earlier of the spindle is going on, these granules are passively following it, and the traces of these movements are seen in the red tracks that are laid down by the image processing software, which is recording the positions and joining them. Remember, the joining process of the tracks becomes important when things are as crowded as this. And Dhruv will, I think, have a little discussion about this when he demonstrates some code to you. Okay. So what did we get from this? Well, we took 3 examples from the database from Marie Delatre's lab, which is published in PLOS Biology. We analyzed the oscillations. We did some time series analysis, got some smoothing. From the smoothing, we did some Fourier analysis, fast Fourier transforms, got frequencies, and then the frequencies of the lipid granules came up to be dominated by 40 millihertz. You see that 0.04 value showing up as a peak. That is 40 millihertz, right? And that corresponds very nicely to previous measurements of the spindle oscillation frequencies themselves. So we can now use this tracking to correlate intracellular dynamics of passive granule movement and active dynamics of microtubule motor spindle interactions. And for this, we have developed a sort of GUI, which allows you to sort of intuitively click things and do things. Now, for the remainder of this exercise, we are going to actually go through a very, very what we call deconstructed code for running this exercise. But before that, you and I are going to have a little session on a basic introduction to Python, which is going to use the Google Classroom environment that I think most of you have signed up for. So I'm going to stop here and ask you, have any of you not signed up to the Google Classroom? And there is a question from Divya Tyagi. I think, I'm sorry, I didn't see those questions earlier. I will answer that in a second. Google Classroom, have any of you not signed up to it? If you can just please answer yes or no. Or if you're confused about it, speak up. You can unmute yourselves. I think you, sorry, raise your hands and I or the co-host or the host will unmute you. Yeah? Yes? Signed up. So has anyone not signed up? Okay. All right. For those of you who have not yet, it's a very simple process. All you need to do is go to the website called classroom.google.com and add the code of the class, which is the following. And I'll tell you in a minute why I would like you to do this. But suffice to say, it's a lot more fun like that, right? Because you can do things. Okay, so here we go. Class code. Okay, this is the class code NVUKMSX. This is how you can join the classroom. All right. Okay, so now I will take the questions. Okay, so I'll go with one by one from what I can see. So I think the first question was from only the left half of the
first
screen
question
is updated.
is from Divya. Do you want me to read it out?
Yeah, actually that's fine. I can see it. I just realized, was there a problem with the display? You could only see the left half of the screen. Is that maybe Murmu said only left half is updating live? Please correct. I don't know what that means. Did anyone have problems viewing the presentation?
So we did not face any problems as such. Just for one slide, there was a lag. So maybe that.
Okay, maybe it was just that. Oh, great. So the question is an interesting one. Does the centroid point depend on the size or area of particles? Can be more targeted according to particles of interest? So it's a good question, Divya, and 2-3 things. Yes, and yes. In other words, so, and you'll see this when Dhruv does the hands-on exercise, because I think that will become a bit more obvious. Right now, this is theory. But as you see these objects here, the inclusions that we were trying to follow, they were approximately 8 to 10 pixels in diameter. Now, the centroid is so we can use either a geometric center of the object or the center of mass, which is weighted by the intensities. You are aware of the difference between center of mass and geometric center. And the choice of the search radius depends interactively, meaning to say as a user, you have to decide that on a couple of things: how fast things are moving, how big things are, and how heterogeneous the movement is. Meaning to say, if there are both slow and fast particles or slow and fast cells in the same field of view, and I would love to be able to pull up one of those. I have a video somewhere. Just a second. You may have to do a little bit of optimization. Now, are there standard algorithms? Well, I mean, this gets a bit tricky. And again, the exercises that we're going to show you later will hopefully point a little bit better. So presumably you see my screen now. If you don't, please speak up or write in the chat box.
It's visible.
Yeah, okay. So this is a figure from Mondal et al. Mondal and company are the lab of Sandhya Kaushika, who's already, I think, spoken some yesterday, isn't it correct? Am I right?
Yes, sir.
Yeah, maybe she even showed this movie. I don't know. We didn't acquire this movie ourselves, just to say. And it's from published literature. It's downloaded from supplementary materials. What you're looking at here are the very fine movements of intra-axonal vesicles as they move along microtubules. Yeah, these are driven by kinesin and dynein motility. And what you should, I hope, appreciate is that they are practically, each vesicle consists of only let us say 1, 2, 3, 4, 5, 6, 7, 8 pixels, and the separation distance between them is also 1, 2, 3, 4, 5, 6, 7. Yeah, so there is a big problem here, because now if I play this, I'm just going one frame at a time. Yeah, one frame I moved. So if you see this to this, it moved almost by 3-4 pixels. Now this is what makes tracking so complicated, because it may be obvious in its concept, it's very simple, but in its practice, it ends up creating all sorts of issues. And we're going to deal with one example of how to solve this a little bit by simplifying the data, which is chymography. And Badir and I are going to sort of work you through it. But anyway, that I hope that sort of answers the question. Can also access the classroom with the email or send to us. Okay, this is, I think, Akshaya is telling you. Okay, very good. Okay. All right. Is there any other questions? Because if not, then I think I should probably take a break here and we will meet at 2 again.
So there are no questions in the chat.
None, right? Yeah.
Yes, sir.
Okay. So what we're going to do is the same thing that we did today in the morning, which is we're going to meet in the WebEx session. I guess, Akshaya, can I use the same link that you had sent me in the morning? For
Yes,
the
sir, you
afternoon
can use
session, is
this
it the same link?
Yeah. Yes, sir.
Okay, good. So we'll meet on the same link. And then what I'm going to do is share my Google Classroom window with you, and so is Dhruv and Badir. And we're going to sort of work our way through it such that while I am demonstrating, you can use your own copy, which means that that code that you modify, you do whatever you feel like, you can experiment with. Okay. So that is the plan for the afternoon session. And we're going to do two kinds of things. We're going to deal with a simplified tracking approach, which uses chymography, which I think there's also discussion later. It's using a workflow that we have published in a paper in PLOS One. You will hopefully see you can read more about it basically if you go back to the paper. And the second part is a detailed description, line by line, of the single particle tracking. So I think I'm going to stop now. And thank you very much for listening, being so patient. And if there are no urgent questions, we can take the rest later. So keep a note of them. Yeah. Okay. Thank you.
I would like to thank Dr. Atle for such an interactive session. And now we will break for lunch and join back at 2 p.m. where I hope all of you guys can access the classroom and we can then get started. Yeah.
Super.
Thanks, Chaitanya. It was very lucid presentation. Thank you so much.
Great, great. Look forward to the afternoon session also. All right. Okay. Bye-bye.
Bye, guys.
See you.